Welcome back to our class. Today we are going to be looking at what we call cloning. Yeah, we have seen that this has been a problem to many grade 12s, how to explain the process of cloning, and sometimes when they bring it in exam, students find it difficult in answering questions. So today we're going to look at the process of cloning, basically cloning of the dolly sheep, and then some of the questions which can be asked about this content. And then you're going to try to answer those questions and also guide you how to answer if it comes to your paper or if it comes in the exams. Don't forget that cloning can be medicinal. It can be cloning basically for medicine. For example, production of insulin, or um, cloning of organisms. Yes. I'm not going to go into the types of cloning whereby you talk about the therapeutic cloning, and then meaning that trying to clone just a part or a tissue, a stem cell, which is going to replace the damaged tissue or organ. And then the other one could be cloning the whole organism. But in this case, it can be either natural cloning or can be artificial cloning. In this case, we find that we use the same type of genome to multiply or to duplicate or to produce identical organisms. Yes. So let's look at what we call cloning. So we are going to explain this process. In this process, you need to have two sheep. The first sheep, let me call it sheep A, and then the second sheep, sheep B. So if you look at our book, the distinction material, you have sheep 1 and sheep 2. So sheep A, for example, if I want to produce the characteristics of sheep B, and I don't want to produce the characteristics of sheep A, so it means that you have to maintain the DNA of sheep B. All right, let's start. So what do you need to do is, you need to do the following. For sheep A, yes, it must be, you must get a cell from sheep A. This is a cell. And then you must also get sheep B, a cell from sheep B. This is also a cell, okay? Cell from sheep A and cell from sheep B. But these cells are a little bit different. Remember that a cell, a full cell, is supposed to have all the content, including the nucleus, and also here. But where do you get these cells? Here in sheep B, because I want the characteristics of sheep B, I'm gonna get what you call a somatic cell. Somatic cell. Why do we need a somatic cell? We'll explain that. But somatic cell means a body cell. A body cell. Don't forget that somatic cells, they have what you call 2N. Yes. So in this case, for example, if the sheep has 62, it has 62 maybe chromosomes, then automatically this is gonna be 62. If it is 1N, it's gonna be 31 chromosomes. So in this case, if you look at sheep A, this one is going to be a gamete. A gamete. In this case, it must be an ovum, meaning that the first sheep, sheep A must be a female sheep, because you can't get an ovum from a male sheep. And sheep B, because I want the characteristics of sheep B, doesn't matter whether is a male or a female. Yes, as long as I want those characteristics. So it means that now I need the ovum. This is an ovum. So the next step is now I've got this ovum, and then I've got, don't forget that ovums are 1N. They're haploids. While these ones are diploids because it's a somatic cell. So I have these two cells. I've explained which type of cells you are supposed to get from the organism. Yes. So because I want the characteristics of sheep B, of this sheep, therefore, I'm gonna keep the nucleus. Why? Because the nucleus contains the genetic material, and then the shell or the the casing of this ovum. So not of ovum, the casing of this cell, I'm gonna throw it out, meaning that I'm gonna throw this out. Yes. So I throw it out, but I keep the nucleus. Why? Because I need the characteristics of sheep B, and there is no way I can get these characteristics except when I go to the nucleus where the genetic material is found. Then I go to sheep A. Sheep A, I want I don't want the characteristics of sheep A. So what do I want? Here, because I don't want the characteristics of sheep A, I will throw the nucleus out and then I will keep the casing. So meaning that now this one becomes an empty ovum. Empty ovum. Remember, this is an ovum. Yes. So it becomes empty ovum. I always ask my students that if I have water, I put it in the bottle of Johnnie Walker, and then someone finds you drinking water from Johnnie Walker bottle, what will that person say? Definitely, that person will say that you are drinking Johnnie Walker or Savannah, whatever. If I have Johnnie Walker, I put it in the cup where you drink tea from. Someone comes in, finds you drinking that Johnnie Walker in a cup. What that person is going to say? That person, I don't think the person will say that you're drinking Johnnie Walker. Will say that you are drinking tea or you're drinking water or juice, whatever. But when you're drinking Johnnie Walker, what does it mean? Why am I saying that? What you appear to be outside is what we take. So what happens is this is an ovum, a casing of the ovum or empty ovum. And this is the nucleus. Don't forget that this is 2N. So these two must combine. So meaning that I'll bring this empty ovum and I'll bring this nucleus. Then I'll put it here using a mild electric shock. We use a mild, I can ask you which method is M. It's called a mild electric shock. So now what happens is, if I have this here, we say that now this is a casing of the ovum. If someone comes in and asks you, what is this? We're going to say it is an ovum. Why? Because the casing is for ovum. But don't forget that the nucleus we used was 2N. When does ovum looks like that? It might not necessarily be ovum, but when does it have this situation? Let's go back to reproduction, just a little. And then we say that, remember, a sperm, yes, has a nucleus, and the ovum has a nucleus. During the process of fertilization, the nucleus of the sperm will enter the nucleus of the ovum, and they fuse. They form, the casing is what? Is ovum. You see, the casing is ovum, but what is in the nucleus here is a mixture of the sperm and ovum. But what's the number of the chromosomes is 2N. So when you go back here, we shall see that this structure looks like this. And how do we call this structure? This structure, we call it a zygote. A zygote. meaning that what we have formed here will look like a zygote. And the moment that zygote-like structure forms, then it will undergo a process called mitosis. So this zygote, the normal zygote, will undergo mitosis to form a ball of cells. Yes, where embryo is formed. And then exactly what happens here is the same process. So here, this is going to undergo a process called mitosis. And then a ball of cells is formed, or embryo is what? Is formed. So the moment the embryo is formed, what happens now? What is the next step? Okay, now I've formed the embryo here. I've formed the embryo here with a ball of cells. Don't forget that you form a ball of cells called the morula, hollow ball of cells called blastula. All that we are skipping all that information. So now you have the embryo here. So what happens before you form the embryo? These balls or ball of cells, we get them and then we insert them in another ship C. How do we call this ship? Because I have to be with a lot of ship C. How do you call these ships? We call don't forget that a ship is a ship, whether it's plural or what, is a ship. So I'm talking about different types of ship. Okay, so you form these different types of what? Ship, but of the same kind. We call them surrogate what? Surrogate ship. So this ship, I'll bring the embryo here. I'll put it there. I'll bring this one. I'll put it there. I'll bring this one. Put it there. I'll bring this one. Because I'm using the same genome, the same genetic composition, yes, then automatically each ship here is going to produce the same kind of ship. Meaning that they're going to look alike. They're going to be like twins. Don't forget that twins are two. Yeah. Or triplets. But they're going to look alike. But you're going to produce thousands and thousands and thousands of these organisms. So what does it mean? It means that now this shape has formed, the one which has formed. So let me say that this sheep is going to give birth to sheep D. You see? So sheep D, because I used the genetic composition of sheep B, yes, sheep D is going to look like sheep B. Why? Because I kept the DNA of sheep D, of sheep B, and I threw away the genetic composition of sheep A, so that I remain with the casing of the ovum. So that when I combine with this and this, I form a zygote-like structure. Yeah, it's a zygote-like structure. Yes. They can even ask you a question. Why is this one not regarded as ovum? Because we use the casing of the ovum. Don't forget that the ovum or gamete are always haploid, but this is a diploid. Yeah, so now when you go back to our distinction material book, yes, is here. This is the process which I was trying to explain, that cloning of Dolly sheep. And remember Dolly sheep, Dolly is a singer, I think in America. They named this after her. So I think, Dolly Petra or something. Yeah, I think those people, now, so that you don't forget this. Anyway, let's start. You have sheep 1 and sheep 2. Sheep 1 is you get a cell from sheep 1. Yes, an adult sheep 1 and sheep 2. You get a cell from sheep 1. Yes, and then you also get another cell from sheep 2. But which kind of cell do you get from sheep 1? Somatic. This must be a somatic cell, meaning that it's 2N. You get another cell from sheep 2, but this is an ovum, meaning that is a gamete. Yes. Don't forget that gametes are 1n. So what happens? You remove the nucleus of sheep 1. It means that if you remove the nucleus of sheep 1 and you maintain it and you throw this casing of the cell, then automatically, since you have kept the genetic composition of sheep 1, it means that you want to produce a sheep which looks like sheep 1. Then you have the sheep 2. You got the nucleus, you remove the nucleus, you throw the nucleus away, and you go with the casing. As I explained to you about that scenario of Johnny Walker and Savannah drinking it in a cup. Someone won't think that this is Savannah in a cup. You say that you're drinking water. And then if you're drinking water in a bottle of Savannah or Johnny Walker, we shall say that, no, man, you are drinking Johnny Walker. Yes. So the casing will determine. So you guys, take note the way how you behave. So the way how you behave will determine how we take you up. We might not see what you do behind or the scene. What we see is what we take. All right, that's how people are. All right, now here you have this nucleus and this ovum. You combine them using a mild electric shock. As I explained that, they will ask you about this method. How do we call this method? We say that the method is using electric shock, using a mild electric shock. So now after that, you form now this structure. This is not an ovum. This is not a gamete. Why? Because this nucleus is 2n. Yes. So it means that it looks like a zygote. Why? Because it is 2n and the casing is the ovum. As I explained it here, that it will look like an ovum. It will look like an ovum because of the casing, but because of the genetic composition, then it's going to look like a zygote. Now we put this, yes, into other ships, different ship, many of them, many of these sheep. Yeah. So that now this surrogate sheep, they will help us to develop the baby. And then now, how is this sheep looking like? It will look like the sheep which gave it the genetic composition. Hence, this sheep is exactly the same as the sheep. As we say that this sheep, why does it look like that? Because it was given the genetic composition of this sheep was used. It's like someone who goes to the shop, does the shop. Yes, you buy a sperm. Yes, you put it into another person, for example, during the proper process. That person is not the owner of the kid. No. This one got the sperm and the other brought the ovum, and then now they combined and they put it into another person to carry this baby. Sometimes you call them the borrow babies. The person who is growing this baby is not the owner of the baby. The owner of the baby is the owner of the ovum and the owner of the sperm. But the baby will look like the one who gave the genetic composition. So that's how we do clone this so-called dolly sheep. Let's go and look at some of the questions concerning about this dolly sheep. Questions are easy. Yeah, now this question came in a different version. In this case, they're saying that this is not a sheep, but the process is the same. It's the stallion. Stallion means it is a horse. I think it is a male horse. When they talk about a stallion, they mean a male horse. All right, the diagram below shows the genetic engineering. That's the process, meaning the process they are talking about here is a cloning process. Cloning. A donor cell was taken from muscle of muscle. Remember, this muscle is a somatic cell of a male champion horse. Yes, you see. stallion to create a new offspring. It means that since we obtain the DNA of the stallion, then we're going to have to produce a baby which looks like a stallion. Cell from the muscle tissue of the male stallion horse, champion horse. And then what happens? They're telling you the nucleus was removed from cell. Yes, what happens here? The nucleus placed in the ovum, an electrical shock result into the ovum starting to divide. Which process? We said that the process is mitosis. The process here which takes place is mitosis. Yes, don't forget that. Yes, because they didn't talk about it. And then they're saying that here the ovum was removed, meaning that they kept the ovum was removed from the female horse. Yeah. Yes. So what is next? Yes, in this case, the nucleus of the ovum is removed and then discarded, meaning that they threw away this nucleus and then they took the casing. While here, they threw away this casing and they took the nucleus. Exactly what I tried to explain. But this is the same question, but they're using they're just changing the animal. This time they would be talking about a zebra. And then someone said, no, sir taught us a stallion. Sir taught us a dolly sheep. It's the same thing. You must not go off. Eh? Yes. So it's the same thing. It's just that you have to know the principles of answering and the understanding of these concepts. So now what happens is this will come in here. Yes. And then when they combine together, remember, they have talked about the method they use with a small or a mild electric shock. And then this structure C is formed. Remember, I told you that this is a zygote-like structure. In this case, we can say that it is a zygote, but it's not the actual zygote that side, but it looks like a zygote because it has all the characteristics of zygote. So the ovum divides normally. They called it an ovum, but in actual sense, it's not ovum. Why? Because of the genetic composition. Maybe they're calling it ovum because the outer or the outline or the casing is that one of the ovum, as I explained to you using the other example of Johnny Walker. Then they're saying that the embryo placed in the uterus of the adult female horse. So now the embryo is now placed into the adult female horse. What happens? Which we say that in this case, we call it a surrogate mother. And then now it starts to develop. It forms the structure of embryo develops. So it is put into this, and then now it develops into another stallion. Why stallion? Because we used the DNA of this stallion. Question. All right. Name the genetic engineering process shown in the diagram above. How do you call this? Cloning. Simple. Process named produced ovum B. Which process produced ovum B? So the process by which ovum are produced, we call them oogenesis. But don't forget that they undergo what we call meiosis, and then oogenesis is being produced as and ovum is being produced. So the process is oogenesis or meiosis because they are not specific. Yes. So they're saying that why is the donor cell extracted from the champion horse? Why is the donor cell? Why did we extract it from the champion? Because we want to produce a sheep which looks like the champion horse. Yes. We want to produce a sheep which looks like a champion horse, meaning that it has the characteristics of the champion horse. The next question is explain why only, they're saying that explain why only the nucleus of the donor cell is used. Why? Why do we use the nucleus of the donor cell? If you come here, the donor cell is When you come here, the donor cell here, when you got it from the champion horse. So they're asking you here a question. Explain why the nucleus of the donor cell is used. Is because it contains the genetic material for the champion or for the donors, for the donor what? Donor horse. Meaning that we want the characteristics of the donor horse. Therefore, we kept the nucleus because it contains the genetic information or genetic material. That is the DNA. All right. Then they're saying that a somatic cell, when you talk about a somatic cell, we are talking about a body cell. And then in this case, it is diploid. In horse, contains 64. Many people, they take this for granted. Some of them, they don't know. Some of them, they always think that all chromosomes in an individual is what? Is 46. It's not. That's a human. A horse has 64. Drosophila melanogaster has 8. So now, so you have to know exactly what are they saying in the question. Don't memorize. Don't memorize. Your head will become big. So don't memorize. You're supposed to use the information they give you in the exam. So how many chromosomes will there be in a structure? Structure A. Let's go to the structure A. Structure A is the, yes, the nucleus. Check. Nucleus of the horse. But remember, this is a somatic cell because it's a muscle cell. Don't forget that here I explained it and I said that this is 2N, diploid. I've explained it. Yes. Yes. You see, it's a diploid, meaning that since it's a diploid and the number of cells they get, the number of chromosomes they give here is 64. So the diploid is going to be 64 and the haploid is going to be 32. 32. Yes. So it means that here the chromosome is going to be 64. And then ovum B, let's go and see ovum B. Where is ovum B? Ovum B is here. Ovum B is going to be the half of 64 because it's haploid. So it's going to be 32. Muscle cell in organism D. The moment they say muscle cell, automatically they are talking about somatic cell. So the answer here is going to be 64. Even if I don't check the organism, but the moment they say muscle, those are somatic cells. As long as they don't talk about the ovum and the sperm, the rest 64. Oh, the rest are diploid. Let's go to check. They're talking about the surrogate horse. Last question. They are saying that explain why the ovum labeled C cannot be considered a gamete. Why ovum? Why is it? Yes. Why do they even call it ovum? Actually, it's wrong. Why is it not called a gamete? They are saying why ovum. The moment they say ovum, it means it's a gamete. So it means that that's a they were supposed to modify that question and looks better. Ne? Yes. Anyway, let's continue based on what they said. Why is this structure C? Yes, I think that would have been the better language. Ne? Why is this structure C not considered a gamete? Yeah, that one would have been a better language for scientists to understand. Why is structure C not considered a what? A gamete. Why is structure C? Why? Because, yes, it's because it contains genetic material is 2N. Ovum cannot be with the 2N. Remember, these are haploids. So it means that since this nucleus is diploid 2N, therefore it cannot be considered a gamete. Hence, I told you that this looks like a zygote. Hence, I called it a zygote-like structure, not ovum. Yes, it's a zygote-like structure. Guys, don't forget that this is a distinction material book. You cannot find any question in any question paper which cannot be answered by this book. use this book from the links below. You'll be able to get a distinction, guaranteed. Yes. And these different colors means a marking point. So the next time we will come and look at the different questions. Let me check here. Let me check here for you. And I see something here. Is there any other question? If you have any question you are struggling with, let me know in the comment section below, or let me know on the WhatsApp. I will be able to help when I get time. Bless yes. You. So thank you very much. I'll see you again. M. Saidi as usual. Good